Start, sir.
Okay. Good morning, dear students. Welcome to another live webinar class of zoology. I welcome you all for this session. Yesterday, we have completed lecture 3 of immune system. So today we are going to study about the lecture 4 of immune system. Okay. So what we are going to learn from this chapter. So far we have completed 3 lectures. I hope you have gone through them. So if you have not seen those classes, first see those classes and watch this. Okay, dear students, let's see what we are going to learn from today's class. Today, we are going to learn about the process of vaccination. Okay, so it is the most prevailing term, isn't it? So in the society, there is a discussion about the making of vaccines and vaccination. So we will learn what is mean by vaccine and what are the types of vaccine as well. Next, immunization. Okay, vaccination and immunization are similar terms, but there is a slight difference between them. I will explain you that as well. Nothing but making a person immune is called immunization by giving vaccination. Okay. Next, types of vaccine. Okay, so vaccines formation, and there are different types according to how the vaccines are formed. Okay. Next, there are some disorders which are caused to the immune system of the body. They are called immunodeficiency disorders. Deficiency means reduction or reduce. Okay, so due to loss of immunity in the body, there are some disorders found. So we will learn about different diseases and disorders regarding immune system. Okay, immunity means you are familiar with it. Immunity means the ability of the body to fight against pathogens or disease causing microorganisms. Next, we also study about the HIV AIDS, and in that we will study HIV structure. It's a virus, and we study about the HIV mechanism and action of how it affects our body. Okay. These are the things that we are points that we are going to cover from this class. Okay, let's get into the class. First of all, vaccination. Okay, what is mean by a vaccine? What is actually present in the vaccine? So what is the most common vaccine you listen many times? That's a polio vaccine, isn't it? Oral polio vaccine. So what is present in them? Actually a vaccine is a substance which is used to stimulate the production of creation of antibodies. Okay, so vaccine helps us to produce antibodies. Okay, antibodies are produced by B lymphocytes in the body, isn't it? In the humoral immunity, isn't it? Yesterday we studied that. Thereby they providing the immunity against certain diseases. Okay, so since the production of antibodies is already done, in future also the disease is prevented. Okay, we won't get that disease again. Vaccination is the injection of a dead or weakened organism. Remember this. Okay, what does vaccine contain? Vaccine actually contains the disease causing organism only. Suppose if we are talking about the polio vaccine contains the polio virus in it, but that virus doesn't affect us. Don't affect us. Why? Because virus is weakened. Okay, it has been killed. Sometimes it is killed, sometimes it is inactivated, sometimes it is weakened. Okay, so weakened organism is present in the vaccine. When this weakened organism is injected in the body, the weakened organism will have antigens on their surface. Okay, since the antigens are present in the weakened organism, these antigens are identified by our immune system. In the humoral immunity, we discussed about the MHC, which is major histocompatibility complex. Okay, they are self-surface markers. What do? They identify our B lymphocytes, macrophages. They identify these weakened organisms and produces antibodies against it. By 7 days, antibodies gets produced, and however, we don't be affected since the organism is already weakened, already it is inactivated. So in next time, if we get the same virus in our body, then we will effectively kill the virus with the help of these antibodies which are formed in our body. Okay. Immunization. What is mean by immunization? Then immunization is the process by which an animal or a person stays protected from diseases. So for the immunization, we carry out the process called vaccination. Okay, vaccination helps for increasing of immunity of a person. Okay, that's why vaccination is a part of immunization. Okay, so let's see different types of vaccines. See how vaccines are made actually. Even coronavirus or any other viruses. So against those viruses and bacteria, the vaccines are produced. How are they created? There are three types mostly. First is live attenuated vaccine. Let's see what is it. Live attenuated vaccine means the pathogens or the bacteria are weakened so that they reproduce very poorly once inside the body and cannot cause any disease, but they can induce a immune response. In this situation, in the live attenuated vaccine, so pathogens are not killed, but they weakened. When they are so weak, when they are so inactivated, when they invade our body, they cannot produce any disease, but their body structure can induce immune response. Since the body structure or antigens are identified by our immune system, our B lymphocytes produce antibodies against this particular virus. Since the antibodies are produced, if any time if in the next time, if any pathogen is invaded in the body, the same pathogen, then it will be killed very effectively by our antibodies because they are already prepared. Okay, so this is nothing but the preparation of antibodies in our body. If the antibodies are already prepared in our body, it can prevent the diseases very easily. Okay, next, killed. Once first method is by attenuating the pathogen, and second by killing the pathogen. In this process, what happens? Pathogens are completely inactivated or killed with a chemical. Okay, so that it cannot possibly produce or reproduce inside of our body or cause a disease, but these also can evoke immune response because their body structure, their surface markers are still existing. They can induce a response, they can help us to provide an immune response, and immune response in turn produces the antibody. So killed organisms also have their antigens on its surface. Okay, attenuated organisms are also have antigens on this surface. Next, third is the subunit. Okay, so by subunit also we can produce the antibodies. How it is done? See, the vaccine is composed of certain proteins, isn't it? Every vaccine and that resides on the surface of the pathogen, and this fragment is responsible for the immune response in the host. Okay, these are the certain proteins, antigens. So antigens, whenever they are injected in the body, against those antigens antibodies will be created. That's called subunit vaccine. These are the three kinds of vaccine according to the organism's ability, according to the life cycle of an organism. We we create these kinds of vaccines. Let's see the types of vaccines. Here on screen you can see the screen, the live attenuated vaccine. Examples for live attenuated vaccines are tuberculosis, that is BCG. Okay, next oral polio vaccine, measles, rotavirus, and yellow fever. For these organisms, for these diseases, to prevent this disease, we inject the live attenuated vaccine. That means the organism is live, but it is completely weakened. Okay. Next is inactivated or killed antigen. So killed organisms, if we are using killed organisms, that called inactivated vaccine. Examples are whole cell pertussis. Okay, pertussis, whooping cough. We call it as whooping cough. Next, inactivated polio virus. Polio virus can also be injected in killed in an inactivated manner. Next, the subunit vaccine. It is also called as purified antigen vaccine. Examples of these vaccines are acellular pertussis, Haemophilus influenza type B, and pneumococcal vaccine. Okay, it's called PCV vaccines. Next, hepatitis vaccine. Hepatitis caused to the liver, and pneumococcal is a lung disease. Okay. Next, the taxoid vaccine. Taxoid vaccine means these are inactivated toxins. Okay, so these are inactivated toxin, means a poisonous substance. Right? So examples: tetanus taxoid vaccine. It is called TT. So it's the most common one, isn't it? Next, diphtheria taxoid. So these are the four types of different vaccines. First is live attenuated, means not killed, it is live, but it is weakened. Next, inactivated vaccine and subunit vaccine. Finally, the taxoid vaccine. These are the different kinds of vaccines. Let's see the difference between vaccination and immunization. Here you see the difference. Vaccination is the administration of antigenic material to stimulate an individual immune system to develop adaptive immunity to a pathogen. Okay, so it is an adaptive immunity, not natural, isn't it? So it's not innate, it is adaptive, and it is because of the administration of the vaccine, antigenic material. Okay, next, the immunization means the process whereby the person is made immune or resistant to an infectious disease. It is actually typically by the administration of a vaccine. So through the process of vaccination, person gets immunized. Okay, so the immunity comes, the adaptive immunity comes by the vaccination of the viruses, vaccination against the viruses. Okay, so this is the minor difference between the vaccination and immunity. Edward Jenner is the father of vaccination. Okay, father of immune system. See in the slide what you are able to see, the baby's vaccination schedule. So up to 1 year, there are many vaccines are administered to the infant. Okay, so why? To be protected, to prevent the different dangerous diseases. So here you are able to see BCG, hepatitis B, oral polio vaccine, triple antigen, diphtheria, tetanus and pertussis, and IPV, injection polio virus. Next, HIV, rotavirus, PCV, pneumococcal vaccine, MMR, okay, measles, mumps, this is to protect from measles and mumps. Next, typhoid vaccine and hepatitis A. These are the different vaccines which are administered in the infant stage, okay, up to 1 year. Okay. Next, Let's see what are the different types of immunodeficiency, immunological disorders. Let's see immunological disorders. And in that, the first is immunodeficiency disorders. Next, we study about the hypersensitivity disorders, and the third is autoimmune disorders. These are the three types of disorders of immuno which are related to immune system. Say, hence they are called immunological disorders. Let's see first what is immunodeficiency disorder. The name itself says, isn't it? So the immune response is reduced. Here you are able to see it is caused due to reduced immune response or absence of immune response. Okay, these are called immunodeficiency disorders. Next, the hypersensitivity disorders. These are caused due to exaggerated immune response to the antigen. Sometimes our body responds exaggerately to the antigens or pathogens. So that's called hypersensitivity. It also produces certain diseases. Next, autoimmune disorders. Auto means self. Okay, self immune. That means our cells attack our body cells only. That's called self diseases, or also called as autoimmune diseases. Unfortunately, immune system fails to recognize our proteins and attacks our own tissues. That's called autoimmune disorders. These are the three kinds of immunological disorders. Let's see what are the immunodeficiency disorders now. Immunodeficiency disorders can be classified into primary immunodeficiency disorders and secondary immunodeficiency disorders. See the slide. The primary immunodeficiency disorders are caused due to defective genes. If there is a problem in the genes, in the manufacture of the gene, or any mutations in the gene, so this will be caused. Example, SCID, severe combined immunodeficiency disorder. Okay, that's called SCID. It is due to the defects that are there in the genes. Next is secondary immune deficiency disorders. These are caused due to infections, pathogens, and aging. So these are the three reasons for the secondary immune deficiency disorders. Example, the best example is HIV/AIDS. Here in the HIV/AIDS, immunity will be lost due to the due to the destroying of the T helper cells. Our immunity is lost. That's why it comes under the secondary immune deficiency disorders. We will study more about these disorders. Next, see hypersensitivity disorders. It is due to the release of such chemicals such as histamine and serotonin. Histamine and serotonin, these are released due to released whenever there is allergic reaction taking place in the body. Allergies are produced by immunoglobulin E type of antibodies, also called these allergens. Okay, immunoglobulin E type of antibodies, they are also called as allergens. So examples are asthma and anaphylaxis. Asthma is mostly seen in the children. It is caused due to the pollen grains and dust particles which are which goes inside of a body. So even whenever the environment is most polluted, these dust particles and the pollen grains of the flowers, they rush inside of our lungs, and they can cause the allergic reactions in the lungs. So that can be called as asthma. So it is a hypersensitivity. People are so sensitive. We call them as sensitive people, isn't it? Because their immune system is so sensitive that they can induce response or exaggerated response to the pathogens or any dust particles. Anaphylaxis disease is caused to the brain. Okay, so in some cases, anaphylaxis disease can lead to the death of a person. Okay, so these are the hypersensitivity disorders. Now let's see the autoimmune disorders. Just now we discussed it is the failing of the immune system to recognize our own proteins. The main function of the immune system is to recognize non-self substances and produce antibodies against them. But if the immune system is failing to recognize which is self and which is non-self, so automatically our antibodies can attacks our own tissues. So that can lead to a severe damage to our body, isn't it? The examples of these autoimmune disorders are Graves' disease. It is caused to the thyroid gland. Due to the Graves' disease, the thyroxine hormone is released in excessively. Okay. Next, rheumatoid arthritis is a joint pain disease. Okay. Next, myasthenia gravis. Okay, so to the muscles, it is caused to the muscles. These are the autoimmune disorders, and Addison's disease is caused due to the low secretion of cortical fluids in the adrenal gland. Okay, so these are the some of the autoimmune disorders. And this autoimmune disorders, some can be applied to the graft rejections also. First of all, you must know what is meant by these grafts. So you have been listening to these new techniques of in the medical field, that there are transplantation of organs, isn't it? So if anybody's liver is damaged, the patient's liver can be transplanted by another person's liver, isn't it? So liver is taken from another person and transplanted into the patient. So that's called transplantation of the organs. Kidneys also sometimes be transplanted. If the kidneys, both the kidneys are failed, what do they do in the family? They will see a match in the family, and they take the donor's kidney and administrate or uh, fix in the patient's body. So that's called transplantation of the organs. So these transplanted organs can be called as grafts. Okay, graft. Here you can see some of the human grafts are cornea. The human eye contain an external layer called cornea. Cornea can also be transplanted. Next, the heart. Nowadays, heart can also be transplanted. Next, liver transplantation is a common one, and the kidney transplantation. So whenever these organs are transplanted from one person to another person, so person's immune system defends these organs. It will not accept the organs. Okay, so against these organs, our body produces immune response. They produce antibodies against these organs, and macrophages, lymphocytes collectively goes to the organ and kill the organ which we are which we are fixed to the patient. Okay, so this is called the graft rejection. Graft acceptance sometimes happen, but graft rejection happen due to the autoimmune disorder. Actually, we are transplanting a kidney from one person to another person to benefit that person, but the patient's immune system is not ready to accept it. It receives it as a foreign particle or the non-self substance, and it starts killing those organs. So this is called the graft rejections. These graft rejections are due to the autoimmune disorder in the body. That's why before the transplantation of the of the before the organ transplantation, doctors thoroughly check the blood typing. Okay, so the blood groups and the relatives and genetic acceptance affinities are clearly checked before going to the organ transplantation. Okay, these are certain some autoimmune disorders. Let's see further. Let's see further about the HIV AIDS. What is the full form of HIV? So we have been studying it from 8th class, isn't it? Today we will learn about the structure of HIV and the process. Let's see. First of all, it is a secondary immune deficiency disorder. Just now we studied. It is an immunological disorder, and it is under the immunological disorder. It is the secondary immunodeficiency disorder. Next, what is the full form of HIV? Full form is human immunodeficiency virus, and the full form of AIDS is acquired immunodeficiency syndrome. What is the difference between HIV and AIDS? HIV is a virus and AIDS is the disease which is caused due to the virus. Okay, so the acquired means nothing but which is obtained. Okay, and immune deficiency, it is nothing but the disease which is caused which kills the immune system of the body. Next, HIV is the virus and that causes the disease called AIDS. What is HIV virus? See, HIV is a virus that damages the immune system, and specifically the untreated HIV affects and kills the T lymphocytes. In the T lymphocytes, there are two kinds of cells, T helper cells and T cytotoxic cells. So HIV attacks on T helper cells. Here you are able to see the picture of HIV virus. Next, ELISA is a test. ELISA is a diagnostic test which is used for detecting HIV. Okay, and ELISA full form enzyme linked immunosorbent assay. This is the disease name. Sorry, this is the diagnostic test name. Okay, ELISA. Okay, let's see the structure of HIV virus. So if you see, it has a viral envelope. Here you're able to see the diagram. You need to draw the diagram as well. Around the surface of the HIV, there is a protein shield that's called, it is made by the glycoprotein. Okay. Next, uh, surrounding the genetic material, there is a capsid which is made by the protein again, and there is a RNA inside of their nucleus. Okay, this is a single strand RNA. Single strand RNA. It's a genetic material. RNA full form you maybe know about it, ribose nucleic acid. Next, main importantly, it has an enzyme called reverse transcriptase. This enzyme is most important to the HIV virus to affect our DNA. Okay, so reverse transcriptase helps for the virus to convert into DNA to mingle with our DNA. How do we get this disease? You can get this HIV disease from all these reasons. First of all, it is also called as transmission of disease. So HIV is transferred from one person to another person by all these four means. First is the sexual contact with infected person. If one person is infected by HIV, if the other person has a sexual contact with him, so it can be transmitted from one person to another person. Okay. So having a multiple sexual contacts is also the reason for spreading of the HIV virus. Next, if the mother is positive, so her baby also may get this disease. It is passed from mother to the baby through umbilical cord. So nowadays, if the HIV infected person is clearly identified, this can be the baby can be protected. Nowadays there are some techniques which can protect the baby from being affected. Even though the mother is positive, we can protect the baby. Okay, so but we must know whether the mother is positive or negative. That's why in the pregnant woman, HIV test is mandatory. It is done, and if she is a positive, then the baby will be protected by some methods of clinical methods. Next is the sharing injecting equipment. Okay, suppose in the hospitals, if the one injection which is used to one patient, if the same injection is used for another person, then through these equipments, the disease can spread, the virus can be transmitted. So that's why we should not use the injections multiple times. One injection for just one time, then it has to be thrown. Okay, so before it has spread, but nowadays all doctors are using separate injections or any materials, laboratory materials to the particular persons. Next, through the contaminated blood. Suppose a HIV positive person is donated the blood. If the same blood is given to another person, so this is called blood transfusions. Through the blood transfusions also, the virus can spread. But the donor should have been positive. If the donor is positive, then only the receiving person will get a disease. If the donor is healthy person, then there is no problem of transmission of disease. Okay, so organ transplantation can also cause the disease. So these are the 4 methods by which the HIV transfers from one person to another person. Next, HIV does not transfer through, and how HIV will not transfer. Suppose if our friend is affected by the HIV virus, HIV patient, if he is an HIV patient, we should not avoid him because it will not transfer through skin to skin contact. We can give a shake hand to them, no problem. Next, it cannot be transferred by hugging or shaking hands or even by kissing. Okay. Next, it cannot transfer by the air or water. Okay. So it is not airborne disease or waterborne disease. It cannot transfer from sharing the food or drinks. We can sit with them, we can have lunch, we can have drinks together, no problem. Share and including drinking fountains. Okay. Next, it cannot transfer through the saliva, tears, or sweat. Okay, unless mixed with the blood of a person. Okay, so saliva and tears and sweat will not contain any blood in it. That's why it cannot transfer through it. That means we can use the towels which are used by HIV patients. There is no problem. We can share a room also with a HIV patient. There will be no transmission. Next, sharing a toilet, towels, or bedding, it will not cause any disease. Next, mosquitoes and other insects can also not, it cannot be transferred by mosquitoes or insects also. So that's why we should not avoid the HIV patients. Okay, so it is, certainly it kills them psychologically. Okay, that's why if you find any HIV patient in your friends group or anywhere in the society, be cordial with them. You can talk to them. You can be as a friend. Oh, first, you must remember, but you must remember how it spreads. Okay, before we have learned, it is only by the sexual contact and some other means. So you must be aware of those, and you can have a friendship with them. Next, let's see what happens. What is the mechanism of HIV inside of our body? So HIV attachment. HIV directly attaches to the T cells of our body. T specifically say if T helper cells, TH cells or CD4 cell, CD4 cells. Here you are able to see this is the HIV virus. So it is binding to the our WBC, and type of WBC is T cell. This is the step 1. This is called binding. Next, in step 2 and 3, what happening? See, the DNA after the binding, slowly the genetic material of the virus is injected into the cytoplasm of our cell. Okay, so in this process, you see the viral RNA through the process called reverse transcription, converting into DNA. Okay, so this is what the importance of the reverse transcriptase. Okay, just now we have seen that, just now we have studied that the HIV virus contain this reverse transcriptase enzyme. This enzyme helps in the conversion of viral RNA into DNA. Why it has to be converted from RNA to DNA? It is to bind with our DNA. You see here, after converting into the DNA, double-stranded DNA, the viral double-stranded DNA gets inside of the nucleus of T cells, that means the human cells, and it mixes with the human DNA. Here you are able to see the viral DNA mixing with the human DNA. So this is called the host cell DNA, and this is viral DNA, and whatever the formed one is chimeric DNA. So which is a viral DNA. So this is how it mixes with our DNA. After uh, joining our DNA, what happens? Let's see. After the latent infection, the chromosomal DNA and viral DNA is combined together, and it and these after combining to it, so there will be the formation of multiple RNAs, single stranded RNAs, and there will be the budding of the organism. So if one virus entered the T cell of our human body, it can multiply into hundreds of HIV viruses. So these HIV viruses again find a healthy cell, again a new cell it will find, and it will go on affecting the T cells of our body. So in that way they enter our body, they enter our cells, they multiply, and they infect the other T cells. Likewise it increases its number using our cells. Okay, so it is like a cellular pathogen, isn't it? See the steps: transcription of proviral DNA, synthesis of viral components, assembly of viruses, and budding of viruses from the host cell. This is how it goes out. It multiply inside of our cell, and it will generate copies of itself. Let's see the life cycle of the HIV virus. Here you are able to see the virus. You can see here it has it is attached to the surface of the T cells, and it is injected inside of our body. So the single strand RNA, through the process of reverse transcription using the reverse transcriptase enzyme, it is converted into double strand DNA. This double strand DNA joins or mixes with the human DNA, isn't it? The human DNA start process of replication, and replication can produce multiple copies of the single stranded RNA, and finally it buds off after the multiplication, after the replication. Many copies of the viruses are formed, and these viruses buds off from the cell surface and convert into a new organism. So likewise, one virus can produce hundreds of its copies, and these hundreds of viruses which are generated, they go and affect a new T lymphocytes. That's how the reduction of the lymphocytes take place in our body. So due to the loss of these T helper cells, our immune system cannot identify the microorganisms or the bacteria. So that's why all bacteria affect our body. Immune system will be degenerated. Okay, so see the diagram. The first step is binding. Second step is the fusion, fusing with our cell. Third step is reverse transcription, converting from single stranded RNA to the double stranded DNA. Next, the integration. So it is integrating with the human DNA. And fifth process is the replication. It is replicating. The replicated molecules are viral DNAs. Next, the sixth is assembly, and assembled in a cell-like structure, and finally it buds off as a new virus. Okay, new HIV virus. Let's see some of the symptoms of HIV. Early HIV symptoms. These are mostly fever and chills, headaches and mood swings. There are multiple symptoms for this because it is due to the loss of immunity. All organs of the body will be affected. Pneumonia, cough with phlegm, shortness of breath and chest pain, high heart rate and low blood pressure, clamminess, rashes, and blueness of the skin. Women, in the woman, there will be changes in the menstruation cycle, lower belly pain, and vaginal yeast infection. So these are the causes we see, symptoms we see. Next, sore throat, swollen lymph nodes, mouth ulcers, muscle fatigue aches, and nausea, vomiting, and loss of appetite and joint pain. So these are the multiple symptoms which are mostly seen in the HIV. Let's see how can we prevent the HIV disease. There are some preventing methods. So it is better, always it is said that prevention is better than cure. So we must prevent this disease. How can we prevent it? By using condoms during sex is the best way to prevent HIV. With suppose if the people are having a sexual contact with an unknown person, it is better to use condom. And condoms can also prevent the sexually transmitted diseases. Okay, there are many other sexual diseases like syphilis, gonorrhea, and many other. So they can also be prevented by using condoms. Next, if you inject drugs, always use clean needle or syringe and never share equipment. Okay, doctors, whenever doctors are using to inject any injection or drugs into our body, drugs means here the medicine. Okay, so they must always use a new syringe for a person. Okay, they should not share anybody's equipment. Not only the HIV, blood can transfer many other diseases. Next, if the person is pregnant and living with HIV, the virus could pass into their baby's body during the pregnancy, birth, through breastfeeding also. So taking HIV treatment correctly during the pregnancy and breastfeeding can virtually eliminate this risk. Okay, so they have to take a proper medical support if they are positive, HIV positive. These are the some of the preventing methods of HIV, and we should not have multiple sexual contacts also. Next, let's see the assignment for today. Explain the mechanism by which HIV multiplies and leads to AIDS. Second, what are the autoimmune disorders? Third is mention various types of immunodeficiency disorders. Okay, dear students, by this we have completed this chapter. So in the next class, we will be starting a new chapter. Thank you. Madam, class is completed. Dear students, not only watching the classes, you have to note down all the points, all the diagrams which are there on a slide on your running notes also. So keep maintaining a separate running notes. As you come back to the school, you have to submit these running notes to the concerned engineer.
Okay, sir.